Hello everyone. Let's get started. Today we're going to be talking about energy, and specifically how energy flows inside of living cells. The way we're going to approach this large, complex topic is that we will first talk about what energy is and why living cells require an ongoing, constant source of energy. We're going to review the laws of thermodynamics and think about how they apply to living cells. Next, we're going to talk about how energy flows in a cell, and that will include a review of oxidation-reduction chemistry. We'll talk about how energy is measured, how it is used, how it is stored, and finally, we will have a brief review of cellular respiration and photosynthesis as we discuss how ATP is made. Let's begin with a definition. The traditional definition of energy is the capacity to do work. When we think about this definition in how it relates to living cells, we might alter it to say energy is the capacity to cause structural or chemical changes in the cell. It's important to remember that all living cells require an ongoing, continuous source of energy in order to perform all of these structural and chemical changes that keep a cell alive. There are several different kinds of work that living cells perform every day. For example, synthetic work, or the work of building new molecules, forming new covalent bonds. Mechanical work, which is the physical changing in position of structures within the cell, moving things around in the cytoplasm, moving the cell itself through its environment. Gradient work, which is the creation and sometimes the maintenance of concentration gradients across membranes. Remember, protein pumps are doing that gradient maintenance work in a cell. Electrical work. There are parts of the body that use electrical current as a means of signal transduction. And finally, the production of heat. Some organisms, like human beings, are homeotherms and need energy in order to maintain their body heat. So, as we discuss energy and how energy flows in a living cell, it's important that we think about the first two laws of thermodynamics. Remember that the first law is what we call the law of the conservation of energy. And the second law is the law of thermodynamic spontaneity. The first law of thermodynamics tells us that energy can neither be created nor destroyed, only altered in its form. A cell cannot pull energy out of thin air, and it can't destroy energy. The only thing it can do is change the form of that energy in order to best meet its needs. You've heard me say before that cells are very thrifty. They don't want to waste. They're not going to waste energy. They're not going to allow it to just be lost to the universe. They also don't want to waste nutrients or other materials. And they don't want to waste the time of doing things that don't need to be done. So when it comes to energy, cells have to be very thrifty. They have to conserve it whenever possible. There are basically three things that a cell can do with free energy. It can either store that energy if it's not needed in that moment. It can store that energy as potential energy. And remember, energy can be stored both in covalent bonds and in ion gradients. Yes, ion gradients have potential energy. It can allow that energy to be lost to the universe, and that would be in the form of thermal energy, heat energy. And it can transform the energy into something more usable in that moment. In other words, towards chemical energy, towards kinetic energy, whatever form that cell needs in that moment. Those are the three options. There are no other options. You either let it go and lose it as heat energy, you store it for later, or you transform it into a form of energy that is usable in that moment. Now, the second law of thermodynamics tells us that in a closed system, spontaneous processes lead to increased disorder or entropy. This is the law that students have a hard time conceptualizing. But when we think about it in terms of a living cell, when we think about the cell itself and how this increasing disorder, this increase in entropy plays out, what it's really telling us is that the universe is pushing towards fewer covalent bonds, smaller molecules, energy being lost as heat. We need to remember that in order for a cell to continue to build large molecules like the macromolecules and do the kinds of structural and chemical work that it needs to do every day, that costs something. That costs the cell in terms of energy. It's working against the natural order of things, or I should say the natural disorder of things. And that takes work. Let's think for a minute about how energy flows in a cell, how energy moves around in a cell. It's important to know that it's the electrons that surround every atom inside of a cell that serve as the conduits of energy. In other words, as the electrons move, so goes the energy in the cell. We've spent a lot of time talking about covalent bonds and the kind of biochemistry around covalent bonds. We know that a covalent bond is simply a pair of electrons that are shared between two atoms. In addition to two atoms being able to share a pair of electrons, we also talked about how sometimes electrons are transferred, with one atom losing an electron and another atom gaining an electron. That was the process behind the formation of ions and ionic bonds. But there's one thing we haven't talked about yet, and that's the idea that inside of a covalent bond, the pair of shared electrons can actually move or shift within the bond as those atoms participate in chemical reactions. In other words, during a particular type of chemical reaction, referred to as an oxidation-reduction reaction, as the same atoms participate in a reaction, in other words, as the atoms in one molecule get reorganized and recombined into another molecule, the electron pair that they're going to share is going to shift in position. It might originate in the center of the bond, and then once the product molecule is made, it may have shifted more towards one or the other atoms. We haven't talked about this concept yet, but this is an important process to understand if you're going to follow electrons in a cell and therefore follow the flow of energy. The important thing to remember is that when electron pairs pairs are sitting in a covalent bond, being shared between two atoms. That is a form of potential energy. That is stored energy. But covalent bonds are constantly being broken in living cells so that those atoms can be rearranged and new molecules can be made. And that means if we follow those atoms from where they sit in the substrate molecule to where they sit in the product molecule, we can also follow the position of the electron pairs. And that's going to give us some information about energy flow. Let's review some of the key concepts of oxidation-reduction reactions. First of all, remember that these types of chemical reactions are very common in living cells. Oxidation and reduction always goes hand in hand. In other words, during a chemical reaction, if we see one molecule or atoms within a molecule getting oxidized, that means that something else in that reaction is getting reduced. Always, always occurring together. Sometimes it's hard to remember whether it's the oxidized molecule that's losing electrons or the reduced molecule that's gaining. It's hard to remember which is which. And that's why we use these mnemonic devices, these memory devices to help us remember. The mnemonic device that I use is Leo goes grr. And Leo stands for lose electron, oxidation. Grr stands for gain electron, reduction. So what is losing the electron or electrons is what's being oxidized, and what's gaining the electron or electrons is being reduced. Remember though, as I said a moment ago, sometimes this is an actual loss, an actual gain of electrons. Sometimes this is a transfer of electrons. But other times, it's a relative loss, a relative gain. In other words, when we follow the atoms from substrate to product, we see the electrons shifting in position in relation to those two atoms. So one atom is going to be partially losing electrons, and the other atom is going to be partially gaining electrons. I know this is a strange concept, but we will look at an example. First, though, let's just remember which is which in oxidation reduction. I can't stress this enough. It's important that you come up with a way to remember whether loss or gain is involved with oxidation or reduction. Oxidation is about the loss of electrons. So in this drawing, they're showing us that this molecule that's labeled A has electrons associated with it, as all molecules do. And through some kind of a reaction, those electrons are being lost. So A is being oxidized. Now, you'll also see in the drawing that they're calling A a reducing agent. Don't worry about that language. That's not important to us in this course. But understand that if A loses electrons, we will say that A has been oxidized. But A does not get oxidized in isolation. If A is being oxidized, then something else is being reduced at the same time. And in the drawing, you can see that that's this molecule labeled B. Any molecule that gets reduced is a molecule that's gaining electrons. Now, A and B could also just be atoms. We could see an atom undergoing a relative loss of electrons, and another atom experiencing a relative gain of electrons. That's the more common thing going on in a living cell, this partial oxidation and this partial reduction. Take a look at the chemical reaction that's depicted on this slide. We can see carbon dioxide combined with water, combined with energy, producing this strange looking molecule. This is actually glucose, drawn in a very unusual way. Glucose and oxygen gas. First things first, do you recognize this reaction? Carbon dioxide, water, and energy producing glucose and oxygen. That's right, this depicts photosynthesis. Now, each one of these molecules has been drawn out with the various covalent bonds, and that's important because along with those solid black lines between the atoms, we can also see the electron pairs. So remember from our discussion of biochemistry, in a molecule like CO2, the oxygen atoms have a higher electronegativity than the carbon atom in the middle. So those oxygen atoms are going to be pulling on the electron pairs and drawing them closer to the oxygen. Okay, this is an example of polar covalent bonds. And because they're double bonds, there are actually two pair of electrons involved on each side here. Water is also a molecule that contains polar covalent bonds because the oxygen is more electronegative than the hydrogen atoms are. So the oxygen is sort of hogging the electron pairs. They are snugged right up against the oxygen atom. Now, this is a very unusual way to draw glucose. Normally we draw glucose with a ring structure. The reason the artist has drawn it this way is because it allows us to follow the atom, the carbon atom, from where it sits on the reactant side to where it sits on the product side. The same carbon atom, in other words. And it's this carbon in the carbon dioxide. This same carbon atom is this carbon atom. It's been involved in a chemical reaction. The covalent bonds that it's participating in have been broken, and then new ones have been built. This oxygen that you can see in the water molecule is the same oxygen as what's over here in oxygen gas. So let's take a look at how the electrons are shifting around these two atoms. This carbon, as it was sitting in a molecule of carbon dioxide, was relatively far away from the electron pairs that surrounded it. We have 1, 2, 3, 4 covalent bonds that this carbon is participating in, and all 4 electron pairs are relatively far away from that carbon atom because they are drawn to the oxygens. But when this carbon has these bonds broken and these bonds built, now it's bonded to one carbon here, one carbon here, one hydrogen here, and an oxygen here. So now 3 out of 4 of the new covalent bonds are not polar covalent bonds, but non-polar covalent bonds. The electron pairs are sitting right in the middle of the bond because carbon and carbon have the same electronegativity. Carbon and hydrogen have the same electronegativity. Carbon and carbon, the same. We still see a polar covalent bond here between the carbon and the oxygen, but these 3 are non-polar. We went from 4 polar covalent bonds to 1 polar covalent and 3 non-polar covalent. What has happened in these non-polar covalent bonds is the electron pairs are now closer to that carbon than they were over here. This carbon is now relatively closer to 3 pairs of electrons than it was over here. This is a relative gain for this atom. This is a relative gain of electrons for this atom. And we call a gain of electrons reduction. Remember GER, gain of electrons reduction. Now, if this carbon atom is being reduced, that means this oxygen atom must be getting oxidized. Let's look at this. As this oxygen atom sat in a water molecule, the two pairs of electrons were very close to it, snugged up right against it. But if we follow that oxygen over here to the product side of the equation, now it's bonded to another oxygen. So even though the electronegativity is high in both of these atoms, it's equally high. So the two pairs of electrons are going to sit right smack in the middle of that double bond. This oxygen had electrons right next to it in this molecule, and now they are relatively farther away in this molecule. So this oxygen has experienced a relative loss of electrons. It's not an actual loss. The electrons weren't transferred. They just moved. They shifted from being very close to the atom to being a little bit farther away from the atom. That's a relative loss. And we call that oxidation. This oxygen was oxidized. This carbon was reduced. The two things always go together.